big data, machine learning. You have no edge at that way. You have no idea where the errors are going to lie, what the correlations are in the data sets that are really as a result of something about the instruments, right? So he's pretty good about that. And yeah, I mean, I'm teaching this course next winter, and then I'm going to bow out for a while and do other stuff. So, but I would, maybe the best thing would be either is to take with Yadatan and Bina, right? Right. So it's a small number of courses. I would really urge you. I would urge all of you to learn statistical mechanics. It's part of physics. Usually they tell you to take all kind of other stuff like basic mechanics and electricity and magnetism, but you really don't have to. I mean, it sits there by itself. And if you know probability well, you could just pick up all the stuff on the side. I mean, that's probably the most valuable branch of physics as far as biological physics goes. And certainly as far as, again, if you're trying to understand living organisms, living organisms are completely dominated by entropy, by random interactions. And the more stat mech you know, the better off you'll be. Just an opinion, right? I don't know why. I mean, there's like biophysics, biological physics submajors in physics, but there's no real neuroinformatics per se. But I think the more you know about where the data is coming from, the more effectively you could analyze data. That's the key point, right? I mean, there's a lot of examples about that. Yeah. Okay. And you guys have any other questions or?
Yeah. So, what specific tools would you advise us to learn if we're going into computational neuroscience?
What tool? I mean, as a theorist?
No, just like practical tools you would use to like analyze, like general data.
I mean, you really want to have rock solid knowledge of linear algebra, period. And that's clear. And I think you also really want to know stat mech. I'm looking at friends who sort of dominate in this field, and these are things they know well. There's a lot of people from computer science who kind of drifted in, but they haven't really left a mark, I would say. For a lot of reasons. I mean, computer science is not science. Computer science is thinking about algorithms and optimization, and there's no reason that biology is optimized. I mean, it sounds good, but there's not a lot of data that way. A few things I get the thing I regret just because it's been painful to learn it is I would really take like a course in topology. I think that would be quite useful. Things like real analysis were a lot of fun. I mean, I did that kind of stuff as a grad student. I mean, as an undergrad, that was fun, but it's one of those. I mean, I could have taken an art class and it would have been a little less painful. So that's, and learning probability theory, which is a little bit connected with stat mech. Okay, I think is very valuable. That's what I'd recommend. All right.
Yeah.
Yeah.
Okay.
Yeah. Okay. Anything else? I mean, yeah. You've got a lot of different opinions from people, but
I have an out of field kind of question. Do you consider the theory of mind? So, developing a computational model for the brain, do you consider that a uniquely theoretical problem, or is that something we can actually do?
Oh my God. Okay, that's heavy. It's not even late enough to drink a glass of wine. I think we just, I don't think there's like there may be some emergent principles. I mean, that's what we all hope. Just like this sounds a little camp, but in physics, it's been emergent principles. Like superconductivity is an emergent property. You wouldn't. It's a very, very weak interactions of electrons moving and tugging on nuclei, positive nuclei, and then by tugging on the positive nuclei, they're actually pulling other electrons, right? Sort of a second order interaction, but at some point it dominates. It's a stinky little effect that you normally would never calculate out. And so maybe there's some interesting effect that ends up dominating, through physics or through chemistry that we haven't seen. But I think we just don't have a lot of data yet in neuroscience. Neuroscience is a really crude subject. We know a lot about how single cells work very, very well. And we know a lot about the how they talk to each other through synapses. I mean, these cause signals to go on only one direction. They have a threshold operation. I mean, there's a lot of plasticity involved, which is really not really understood. And that's a big difference from electronic circuits, right? Electronic circuits are not, you don't think of them being fundamentally plastic, but the connections, like what would be the equivalent of a field effect transistor on the input to a circuit is changeable, but not continuously changeable, or it would be like we were on morphine and not remember what we said, 10 seconds ago. So I think we're not quite there in terms of data, right? I'm kind of under the feeling that a lot of times you only understand things when you collect enough data to get that it's accurate enough and it's large enough, and then all of a sudden some simplicity happens, right? Mm-hmm. So I mean, we were lucky, right? This happened to us just this last year. We've been messing around in the vascular system for years, and there were all kind of ideas that go back to 1919. That's a long time ago. That's 102 years ago, right? About just how you do something almost trivial, like relate vascular density to energy use. But until we finally had enough data, you couldn't say anything intelligent. And then you get enough data, and then you remember your grad school level stat mech or senior level stat mech and solve a diffusion equation by hand, and you say, whoa, it makes sense, right? So I think we're just we don't have enough data on systems in neuroscience. We have very little. Mm-hmm. So we're not there. I mean, it's great to think, you want to understand the mind, you want to understand how, well, frankly, how matter came together to try to understand itself. It's a totally outrageous concept. Mm-hmm. But I don't know we're there yet. I mean, maybe other people have very different opinions. I mean, cognitive, there's a whole cog sci department which thinks about this stuff, but I
think has the current predominant hold is, I think it's Ray Kurzweil's theory. It's a really crazy semantic web of hierarchies. And again, that's a purely computational construction. There is not much in the way of, I guess, physical or chemical evidence to support that claim. But it to me at least is just incredible that the human brain operates on 20 watts and is better than any processor will ever have.
Maybe not.
Okay, for at least the next 50 years.
Okay, then it's not my problem. 50 years from now, it's my daughter's problem.
Fair point.
Your problem.
Fair point.
She's kind of your age. Yeah. I don't know. I mean, this stuff is kind of fun to talk about it. Maybe you sort of have to talk about it because you have to think about how to address these things experimentally. But I'm not. I don't know. Psychology was kind of rich, and now it's kind of. It sort of lost some of its flavor, and it sort of fell back to looking at fMRI images and just which is basically using the oxygenation levels in the brain to come up with, models of thought. That just seems, I don't know. That just seems a total waste of time. It's not my gig.
Yeah, I'm with you on that. I find that looking at the brain from outside without employing some crazy amount of computation only is really like taking a picture of atoms and trying to make inferences about how they move.
Well, they do, okay. So actually, so my friend in grad school, Don Eigler, was not the first person to image atoms. That was Binning and Rohr. So when I was in grad school, this picture came out in PhysRev letters. It was the first tunneling microscope picture of individual atoms on the surface, right? So these guys had guts. I mean, they took the first picture of atoms. Everybody thought the vibrations were going to kill it, but they weren't doing the averaging right, and they basically published one paper, and once it was out of the bag, everybody started copying it and started doing it better than them. But that's what the Nobel Prize is for, right? For the first idea. But I had there were three of us who left. I was a grad student here. There were three of us who were here as students, and so we went out east to Bell Labs, which was still running at that time. And Don had a terrible time there. He didn't like his advisor, his postdoc advisor. And then he drove his somewhat souped up Volvo from New Jersey to, he went out, he got a job at Almadine at IBM. He drove it in like 3 days by himself, which I happen to think is a little crazy. But he went on to make this little corral of atoms, and then he went on to write IBM and atoms. It got on the cover of Newsweek. It was very impressive.
Yeah, anyway, thank you so much. Some great stories.
Okay, any, all right, so you want me to show a little bit of what we're doing? What's the scheme?
Yeah, so generally we have professors just talk about their work, maybe like share some recent publications, and generally describe what not just what your lab is doing, but what are the implications of your work.
All right, I'll show some pictures, but I want you guys to interrupt me a lot, okay? And unfortunately, I didn't bring my, I bought one of these Apple pads that have a pen, so I didn't bring it today. They're actually, it was actually a very good teaching tool, but it's a ripoff because it doesn't give you access to the directory structure in a simple way. It's not fun, but all right, anyway. All right, let me go find this thing. Somewhere, come on. Okay, so I'm going to share the screen. Is that cool? All right, but that's not an excuse to turn off your picture and fall asleep. Okay, so can you see this guy? Everybody?
Yeah, we can.
Yeah.
Yes.
All right, so let me explain what happened. Years ago, I'd worked in condensed matter physics, and I got interested in neuroscience and in biological physics. And I heard they started a sort of small neuroscience group at Bell Labs. And at the time, if you're in condensed matter physics, I mean, that was the place to go. That was like that's where the cool kids went. And okay, so things were puttering along. And then at one point, with a colleague, a friend, a guy named Kerry Delaney, we had a lot of fun measuring like electrical waves, just propagating fronts. It wasn't clear what the biological impact of this was. It was almost like physics within a neuroscience setting. So this is going back quite a ways now. And but at some point we tried to push our measurements of looking at these large patterns of electrical waves, right? Like water waves, just but of electrical activity running through the brain. We tried to push this into rodents, and which of course have a bloodstream and in their brain. And we had all kinds of noise, all kind of problems. We couldn't make a clean measurement. And it turned out that the individual red blood cells were just adding noise. They were blocking the light going in that we were using to image. They were blocking the fluorescent light coming out. We wrote a very boring paper, although it's referenced a lot, but it's really boring as all hell. And then this other friend, about a half a year later or so, another friend of mine, a guy named Winfried Denk, who was probably the best and the brightest of all the people from physics who went into neuroscience, had invented this technique, this nonlinear imaging technique, where you use extremely high intensity pulses of light. I mean, so high that, in fact, you could cause the molecule to absorb 2 quanta of light. So you satisfy angular momentum conservation, you satisfy energy conservation. And without going to the details, this allows you to sort of penetrate somewhat deep into tissue. And it really sort of changed the world of cell biology and neuroscience. Anyway, he just said, why don't we just look at the blood flow, right? So, you know, this was a nice place to work, Bell Labs. You didn't teach, you didn't have to raise money. You know, people were smart, it was great. And we found that we could actually measure individual red blood cells in the brain. And somehow this just took off over the years. And so let me just describe a couple of things, because the vascular system is kind of interesting in the brain. It's a fully connected graph. Every node has the same degree. Things meet as triplets. It's degree 3. Because it's has constant degree, you could, not me, but others have proved some theorems that about how the graph breaks up, some percolation theorems, and you can start to see a lot of these effects even in real animals. So it's a nice interaction between math and biology. Okay, so this is a pretty picture of a mouse on drugs that has nothing to do with reality, but it's anyways, to amuse children. Okay, the other thing about a lot of modern science is the techniques, experiments are not showing any of the apparatus. I mean, so I joined forces with a computer scientist, Yoav Freund, for those who are in computer science. Difficult, okay, very smart, but difficult, okay? And, it takes a lot of people and it takes a lot of equipment and it frankly takes a lot of money to pull off these experiments. I think if people know how much it costs per scientific paper, they'd shut down the university. I mean, it's a little outrageous. Okay, so let me just give you a little bit of an idea. So this is some recent work, and we asked a simple question. So for many years, we just wanted to build a roadmap of the brain, right? And I'm gonna, I need to get, oh, here we are. Okay, I need to get a little annotation tool. Okay, so my handwriting is really gonna be poor because is this gonna work? Yeah, okay. So, I mean, it's kind of interesting. Your brain, as somebody pointed out, runs at like 20 watts. You know, the mouse brain even runs at a fraction of a watt. And, but you really have to get oxygen into the brain. You have to get glucose into the brain. You have to get blood flow that's going through the brain that's being replenished constantly. And the flow has to be distributed uniformly throughout the whole brain. And how you do this wiring seemed not so obvious. So we decided already like, you know, sort of like 28-ish or so, or earlier, actually, maybe more like 24-ish, to try to take a stab at this. And so you could think of just slicing the brain up into, you know, thousands of little sections and then, you know, mapping where all the blood vessels are. And you probably could hire enough people on the planet to help you do this with everybody looking at one picture, but you go crazy. So we cooked up a lot of automated schemes. I'm not going to show them here. It's just way too much. But basically, you can if you have light, right, like with a microscope, he could resolve, maybe about a third of a micron in this direction, right? Micrometer. This way is a little worse, okay? Because the light, comes in anisotropically. It shines in from one direction. So just some geometry, this is a little worse. Even spending a lot of money and doing the best you can, you're maybe about 3 microns. So just trying to use light to look at a sample, what'll happen is, if you suppose you have a round tube or something, right? If the tube is coming like straight at you, it'll look like this because things are symmetric in the focus plane. But if that's not true, if it's not like looking right at you, but it's perpendicular, it's going to be distorted. It's going to look like this because of this anisotropy. So before I mentioned, like, it's good to know the physics of your measurement, right? Because you might see this and you might say, oh, it's real, right? They had ellipsoidal tubes in the brain, but it's not true, right? It has to do with the measurement process. So, we know optics pretty well, so we could actually correct for all these things. Okay, so here's the first lesson. So we started this and we built some technology and we did it, and then we basically reconstructed a little piece of the brain, and we published already about 8 years ago, 2013. And then we just got exhausted. It was painful and tiring, and the postdoc got a job, went to Israel, and most of the data was taken in the summer, and the power failed twice, and we lost data, and everybody was just tired of it. So we said, we're going to take a break. We did other things, and then we came back, but then we had competitors. And the competitors said that they're going to not respect the physics of this, but they're going to use convolutional neural networks to actually help decode the vasculature. So their data really sucks. And they got all the dimensions wrong, and they couldn't do any analysis on it. But this was like a known problem. You put a tube, well, I'm not drawing it well, through an illuminated tube. I mean, this has been known since Maxwell. It was really worked out by Max Born about in the 1950s, and you could do all these calculations. So think about the physics or the biology or whatever before you think about the analysis method, all right? Some things can be solved exactly, okay? All right. So that's my piece of advice. Somebody says, I want to use a neural network, I won't talk to them anymore. Okay. All right, so what do you get out of this? All right, so this was actually, if you guys are interested in any of this, I mean, this stuff was kind of, is published, and it's on our website, and I'll go like sit with you and talk about it. I just want to bore you now. Okay, how do I get back to the annotation tool? Okay, clear. Okay, so what do you get out of this? So we had all this data, and we had there's a mouse. I mean, they're tiny, right? But the little mouse has 6.5 million blood vessels in its brain. And we mapped every one of those, and we got the size right. And you could look at that then as just all these tubes, and all these tubes are sources of oxygen and sources of glucose. So we could calculate, numerically, solve the diffusion equation, find out how things diffuse, find out that how, like, the worst case distances away from a blood vessel is related to the density of the vasculature. You could do this kind of mixed numerical and analytical solution that really pays off at the end, because, like, this little red guy is the fine blood vessels. It's a capillary. It's coming right at you. It's sticking out of the board. This guy, and you see my pointer? This sort of swath here, that's a capillary kind of almost in the plane. And we can just calculate what we expect to be the oxygen. And we could derive a little equation, even, makes us feel happy. And rho is just what we call the vascular fraction, what fraction of the brain is filled with blood vessels, okay? There's a couple of other constants in here. I've sort of brutalized things. But then back in the 1980s, right? So, like when your parents were, well, they're from high school or something, it was very fashionable to measure energy consumption. And today you could do this with even with, well, how would you do it today? Back then it was done by having animals have a radioactive glucose, and then you'd slice up their brains, or today you could do it with positron emission measurements. But there were measurements of how you were burning up energy as a function of where you were in the brain. And we had measurements of density of vessels, you know, just an average over all these details as to where you are in the brain. And then we could plot them against each other. And those are all the little blue guys. And these curves are calculations from, and what you find is this simple relation on average kind of explains how nature sets the density of the vasculature according to how you use energy at rest. Okay, so when you start thinking hard, when you take your algebra, you know, whatever, your algebraic graph theory class, and parts of your brain heat up, that's not contributing to this. But just when you're in bed, being mindless, white matter uses this much energy, cortex uses this much energy. Inferior colliculus is responsible for hearing. So hearing is like the last sense to go. Okay, that's why people used to get pissed off when somebody next door has the radio on too loud. Okay, but you really can use your knowledge of physics to derive an equation, and then you find out that the data matches pretty well. Okay, so this is kind of one fun example. We have one more step to do. It turns out that if you go back, let me get out of here. Sometimes you don't know where the hard things are until you make a measurement, right? And let me go down. So we thought it was going to be just a real killer to measure all these little vessels, right? Well, I mean, it wasn't trivial. It took us about 2 years of Shang-Chi's life, okay? But it turns out that a lot of the big vessels are actually built into the bone, which, not to sound like your parents, but this is a good reason to wear a bicycle helmet. Okay, you don't really want to crack your skull. It's bad, okay? So now we're back to doing some technology so we could get all the surface vessels and then really get the whole flow pattern. And I think this was kind of fun because it looks like, you know, within a lifetime, you could go from nothing to something. Okay, so where's a little bit of physics come in, or a little bit of math, I should say. So do you guys know from percolation theory? Anybody know what that means? No? Okay, so, you know, fracking became very popular. It depends on which side of the economic divide you're on. Either it was a great thing, it made the US sort of energy independent, or it's a bad thing because it's, you know, maybe ruins a couple of water supplies. But it actually came, but the idea came from math. It came from this thing called percolation theory. And it's the idea that you have like a solid stuff, like a rock, but then there's cracks in the rock. And finally, the density of cracks gets so high that you could perfuse through a liquid. Okay, so when you could think of, in an abstract way, all these blood vessels as, you know, they're all kind of short. You know, I mean, they never get above, in the mouse, 50 or 100 microns. So if the density gets too low, it's a break, right? You'll never get flow going through. So you think there's some point, if you start to remove blood vessels, the flow is going to shut down. And that's what you calculate. So this is a prediction, and not we, but some collaborators in Boston and Charleston are looking at this. But you start to remove blood vessels one by one, and nothing really happens. You know, nothing really happens. And then all of a sudden, there's this sort of sharp transition that's called the percolation transition, where the flow just stops, okay? So the impact or the prediction for animals is that you lose a capillary, you know, you get hit a little too hard in a soccer game or a rugby game, and it doesn't matter. But too many hits and you start to lose too many capillaries, and then the blood flow just deteriorates and collapses very, very quickly. So what probably happens in aging, you're losing neurons, you're losing blood vessels, and then you just collapse very, very quickly. So again, I mean, I think this should be in the world of bioinformatics, right? It's really data analysis. Anyway, the NIH loved us for this. They gave us a lot of money, so here we are. Okay.
So a quick question, Professor.
Yeah.
So based on this experiment, I'm a general biologist, majoring in general biology. So when you say, I notice the majority of your test subjects are mammals. So this also applies to say the other phylum, say tortoises, to crocodiles. Can we even apply this to older extinct mammalians, or is this only in
Yeah. We're not mammalian centric. So there's this thing called the vertebrate nervous system plan. Okay, and I mean, okay. So somebody asked about the theory of mind. So not to digress, but there's this plan of nervous systems that's sort of been adopted by all vertebrates, and it didn't have to be this way. It just is. I mean, somehow it dominates, and it's birds, turtles, mammals. I mean, you know, individual structures may be heavily accentuated in one vertebrate over another, but the basic plan is there, right? They all have blood systems. And actually, there's a lot of vascular work that goes on in zebrafish. Even on a zebrafish paper, right? That's going to come out soon. I mean, it's a very clear, a juvenile zebrafish is very clear. It's like it's an optics jock's dream as far as imaging goes, okay? And so these ideas are general. I mean, when you get down to, you know, out of mammals or out of vertebrates to, you know, to mollusks, or the game really changes, right? Or encephalopods, like a cuttlefish or an octopus. They have a very different plan to their nervous system, much more distributed plan. They have sort of a different evolutionary basis. It's mostly from modification of message RNA after it's before it's transcribed. I mean, it's so people think their evolution was slower because it's most of the differences in protein are due to what's called post-translational modification. I mean, it's kind of fun, right, to understand why different strategies seem to be more successful than others, right? Yeah, but this would apply to a turtle. We just, the reason we use mice, I mean, in large part, I mean, as opposed to say a turtle or bird is there's a lot of transgenics that's been done on mice. So there's a lot of weird animals you could get a hold of that are really critical. Okay, that the same principle should hold across animals. So why not do it on a mouse that potentially maybe has some medical impact, right? That was never our interest, but it turns out that it does. So, you know, why not? Does that answer?
Yes, I think it gives me a good direction. Thank you.
All right, so I'm really going to scoot along here. It's just the last picture I'll show. This actually comes out of an older paper from 2013. How do I get rid of this guy? So Phil Tsai is now a teaching professor in physics. Maybe some of you might have him someday. I don't know, for Physics 1. It was a lot of fun. Phil was about 25 and Harry Sewell was actually 90 when we published this paper. So it's a huge spectrum of ages. Harry was sharp as a tack, but just to give you a little bit of a flavor of the density, I mean, here we actually made some pretty pictures and reconstructed the vasculature. It forms all these closed loops. It's really kind of, the topology is kind of fun, right?
Dr. Kleinfeld, I have a question.
Yeah.
About that picture, what does the word pile mean? Is that related to the number pi?
Oh, no. Okay, yeah, no, not pi, yeah. Wow, okay, a biologist who knows pi. Okay, so, no, peel is just another word for surface. The pia is this sort of connective tissue that runs over the surface of the brain. There's a dura, which is thick, and then there's this other sort of thinner layer called the arachnoid, and then below that is the pia.
Oh,
Yeah,
I get it. Yeah.
Anatomy will kill you, but so this just means surface, right? It's like a fancy, it's it's like a 50-cent word or something. Yeah. Okay. All right. Yeah.
So when you're modeling this, you said like there's a major emphasis on topology, because I feel like it would really suck if this was modeled based off of a XYZ axis. Are you modeling all of these relative to themselves, or is there like some representational organization with topology that makes this intuitive, or how do you guys do that?
I mean, the topology is just that wherever you look, right, you see these guys meet as triplets. I mean, that's basically the same everywhere. It's all triplets. It's all short range connections. The percolation limit, which is really a function of topology, is the same everywhere. Okay, that's
okay. So you use scale and rules, like complexity science.
Yeah, you could put it that way. I mean, so what happens is at the end of the day is you have this, and between any 2 points, I'm gonna go back a picture, like here. I mean, it's like a little snippet out of that thing. I mean, we could look at the length from here to here, the absolute length. We could look at the mean, the sort of the median diameter. From that, you know what the resistance is. I mean, the flow doesn't really change that much in your brain between active and inactive states. So, linear modeling goes far. So then you're basically writing down this as a graph. And if you write it as a graph, then you could solve it. It's just an electrical circuit in 6 million dimensions. But I mean, that's easy these days. There's nothing special. I mean, you spend more time finding the bugs in your code than having the computer calculation run. Yeah.
Thanks.
Yeah. Okay. So, yeah, a lot of these details, I mean, people worry about these things, angles, tortuosity, but it's not important. It's not important. Okay, I'll do one more thing and then we're going to split. So everybody knows from fMRI? Yes? No? You sit in a magnet and somebody's looking at a monitor and they can tell you exactly what you're thinking. It's not true. But so this was actually the so-called bold effect, that you could actually use MRI, magnetic resonance imaging, to tell brain state. Was a discovery by Seiji Ogawa, who is worked at Bell Labs. I mean, just at the time I got there, he made this discovery. It's another one of these serendipitous findings. He was sort of teaching himself how to do MRI, and the oxygen line to the rat froze up, and he was just watching the signal change. And I think most people would have just shrugged and walked away, but he said, "What's going on?" And he figured out it was the oxygen line. And Seiji knew that when hemoglobin binds oxygen, it has a very different magnetic property than when the oxygen is stripped off. And that just led to this discovery, and it kind of took over the world. And then let me see if I could go on here. How are we doing on this? So another thing that's actually very interesting is the dynamics in the brain. So I mean, you all learned back in high school, you have veins, arteries, capillaries. So if you look at the arteries in the brain, which were these things on the surface, go back, these guys, the red guys, right? They're all hooked up to each other. They form a lattice, right? They're all interconnected. I mean, this looks like Boston, okay? Or it looks like the power grid in the US, right? It's all interconnected. And then not only that, but those arteries are oscillating. So this is one isolated artery that this guy, George Oslo, isolated, ligated, pressurized, brought up to the right temperatures. It's not that easy to do this. And they're just sitting there. So your brain is filled with these oscillators that are oscillating at 0.1 hertz. That's about 1/10 of a second, okay? And people kind of didn't pay a lot of attention to it, but we knew about this stuff. And then, okay, so this is the guy who discovered the fMRI signal, Seiji. Camille Ogilville runs an fMRI lab in Minnesota, best lab in the world. Partha Mitra is my very brilliant, very difficult friend. And they published this interesting paper a long time ago, and they looked at various parts of the human brain, and they're all kind of oscillating around this frequency, around 0.1 hertz, but different regions are slightly different. And then, so yeah, around 2015, I get this email that these guys, David Van Essen, Camille, that they were going to have an article the next morning in the New York Times, that they found out the human brain had 100 more centers than people realized. It's like in the Wizard of Oz. It's like the wizard gives you a piece of paper saying your intelligence has gone up, right? So, and they say, could you read the article and say something pithy by tomorrow morning? All right, so I took out a glass of wine, I read the article, and they basically used these oscillations and found out that different regions were at slightly different frequencies, and that's how they counted different parts of the brain. Okay, so that was great, but it kind of stuck with me, like, what the heck is going on? Okay, so I'm gonna zip ahead, and this is sort of dominating a lot of our thought at the moment. So I forget all this crap. I mean, one thing that happens, this is data from Celine Mateo in our lab, is that I have to get the right picture here. Let me get the right picture. Okay, the right two pictures. Okay, here's a picture. All right, this is summarizing a lot of data. So you have that there's a lot of different flavors of neurons in your brain. And one flavor are cells that try to inhibit, turn off all the other cells. And if you take a bunch of cells and they're each trying to inhibit their neighbor, what happens is they all turn off together and then they all recover together and they oscillate up and down. And they do this 40, 50, 60 times a second, right? It's called the gamma rhythm. If you guys are reasonably awake at the moment, your brains are doing this, okay? And then on top of that, the amplitude of this oscillation is very slowly varying at that 1/10 of a second. So what Celine showed is that this slow variation in this high frequency power, right, in the so-called envelope, okay, is actually can drive those oscillations I showed you before in the blood vessels, and that's what you pick up with MRI. Okay, so this is a fun fact, and okay, so I don't think I have any pictures? No. Okay, so then I'm going to go on. So we had fun and we pushed on this, and then we have a kind of interesting viewpoint. Having trouble. Here we are. Still having trouble. Come on. One more. One less. Okay, I'm not touching anything. All right. This is our idea. So each of these arterioles in the brain is its own oscillator. And it's seeing all these competing forces. It has neurons underneath trying to drive it, maybe at a different frequency than its own natural frequency. And then it has neighboring blood vessels, oscillators, that are talking to it. Okay, and I didn't tell you how, but they're talking to it. And they're trying to make all one frequency. Everybody's trying to make one frequency, but then the neurons underneath are trying to pull things to their own frequency. So it's a nice competition. And it's interesting because this is the kind of thing you can make a mathematical model of. And this was done. Yeah, so let me just give you a little picture of how this works. I think the, all right, I'm gonna try to go full screen. You guys tell me if you can still see this. Do you see the picture? Yeah? Okay, do you see me changing the picture? No, I was worried about that. Okay, so many years ago, 84, right? Bart Ehrmantraut, very funny mathematician, Nancy Kopel, very serious mathematician, published an interesting paper where they just took oscillators, pure oscillators, and they coupled them. And then they changed the coupling constant. And then each oscillator had a different frequency. And what they showed is that as a function of coupling constant, you could actually get these things to break up into parcels, right? So that struck us that we could do this in 2 dimensions. And we could have a grid of oscillators. And they're all trying to now pull each other to some average frequency, but maybe they could just break up into different groups. All right, so that's math. But let me show you what happens experimentally. It's kind of cool. So forget all this, forget all this. So what we did here is we're looking down at a brain, and this is work by this guy, Tomas Borghini in the lab. And we could drive the imaging, the visual part of the brain, that's here. Let me get rid of this guy. It's called photic. And we could drive the whisking part of the brain. So this is the brain responding to light. This is the rat's brain responding to touch to the vibrissa. And what happens is this region here is at the frequency we're driving the light, and this is the frequency we're driving the whiskers, but it spreads. If we could do something interesting, and then we could flip it. We could just flip the frequencies, right? Now watch something subtle here. This whisking frequency is much closer to the natural frequency of the vessel's vibrations than the photic stimuli. So the vibrissa one is really dominating. Oops. And now when I drive the eye at this frequency that's closer to the natural vibrations, the territory it takes up grows. So somehow we're beginning to see some competition. And even more so, we saw something weird. We're going like this. Stim, it's like a pulse, right? So you get all the harmonics. So sometimes we see the blood flow, just oscillations lock at twice the frequency we're stimulating at. So then we thought this is great, and I'll just leave you with this. We can have a little bit of fun. So we have this idea that from coupled oscillator theory in math, that what the vessel wants to do is, if it has competing inputs, it wants to lock to the one that's closest. So if the natural frequency, we just call it F basal motion, FB, is closer to the stimulus frequency or closer to twice the stimulus frequency, it'll lock either to the stim frequency or twice the frequency, right? It wants to make this difference smaller, right? You just want to lock to whatever closest. And because we're stimulating the same system, the whiskers, the coupling constant should be about the same in both cases. So we're going to do the experiment. Okay, the writing's a little small, but basically, I'm going to go to all kind of different natural frequencies, and I'm going to drive the whiskers at all kind of frequencies. So that generates, of course, the fundamental and the harmonic. And I could ask if the locking to the first harmonic is preferred, gives you a smaller difference as opposed to locking to the second harmonic. All right. So let's just see what the data looks like. So this was a lot of fun. This was actually a couple of months' work. So this is the neuronal activity at the second harmonic over the fundamental. So it's a log scale. It has to be, right? This is 1, 3, 10. This is a third, a 10th, a 30th. Okay. And the red dots and the blue dots are what the vessels are doing. So one is if the vessels tend to lock at the second harmonic, and the other if they tend to lock at the fundamental. Now, what the vessels do should really have not depend on what the neurons do, and the distributions aren't completely overlapped, but so it's not something's going on. But if we look at what's happening to the vessels when the fundamental frequency is close to the natural frequency, then most of the dots are on this side, and you tend to lock to the fundamental. And when the harmonic twice the fundamental is closest to the natural frequency, FV, then you tend to lock to the second harmonic. So, I mean, think about it. I mean, this is a mouse and a blood vessel, and it's just totally complicated, icky biology, and some calculation out of coupled oscillators that basically strips off the amplitude and just looks at the phase of how oscillators interact with each other, gives you something that's not too bad, okay? Not too bad. So this is what we find to be fun. You can, sometimes nature does simple things. Okay. So that's just supposed to like tease you, to tell you that, knowing a little bit of physics and a little bit of biology could be dangerous, but it could also be a little bit fun. Right,
I had a question.
Yeah,
Wait,
yeah.
I had a question about the connections part. Could you talk to us a little bit about how, if it's possible, how I guess like regeneration of these connections, say after a little bit of like brain injury, or like how you can make new connections between the blood vessels?
Oh, wait. Okay, you're asking something useful, which I don't know anything about. You really that's. I mean, there's some evidence for regeneration of capillaries. You certainly in the adult don't get regeneration of arterioles. It looks like there's evidence for, vessels splitting, not splitting, sorry, vessels actually forming a junction, okay? This is probably what's happening during development. You form very large vessels and then you form junctions between them. I don't know how this occurs, okay? And I don't know if it's that well understood. I mean, I could just, people say it's neurotrophic factors, but those are just words. They don't really know. But I don't really know anything useful, and I kind of defend that position because, like in the course of these studies, like we found out what the impact was of affecting one blood vessel, and the neurologists at first gave us an amazingly hard time. And unfortunately, we were kind of funded through bioengineering. And then all of a sudden, the neurologists realized it was important. So then, like the National Institute of Stroke said, "Oh, this is great." But I mean, for us, it was just an interesting way to understand fundamentals. So I'm not. I think if you have an opportunity to understand fundamentals, you should do it, and practical things will come out of it, right? And if you're worried about practical things, you're never going to. You're always going to be chasing, some phenomenology. I mean, you're too young to worry about practical things. You should worry about abstract things. I'm serious. I mean, that's just my feeling. Okay.
Okay.
I mean, probably not.
You mentioned.
Sorry.
You mentioned neurodevelopment. I had a project last quarter where we actually worked with like organoids to see how neural activity sort of corresponded with gene activation. When you're talking about these competing networks, have you like researched this from a neurodevelopmental perspective, or where did that come from?
Yeah, I mean, that's a fair point. So we haven't. I mean, it's something we'd like to do. I mean, so the reason for this is that so far these experiments have been painful in the sense they've been very involved. And so like the stuff I showed you at the beginning by Shang-Ji, I mean, when you get down to it, each brain took about 10 days just to take the data and, probably a month to analyze. I mean, there's no reason we think, you know, with X-ray scattering, we should be able to get it down to an hour and then maybe about a week of data analysis, but data analysis, we don't have to actually be looking at stuff. It's just running. So right now it's just a little too expensive, but we're sort of pushing towards being able to do this on a more regular basis. And then you could ask these questions. I mean, right now we just wanted to get even just a couple of samples of quote normal adult animals and get some answers, right? And then we could sort of get answers on diseased animals or animals that may some genetic mutation that changed the nature of the vasculature but maybe preserve flow. You know, just try to get some wacko geometries and see if we could learn something interesting from it, and also look at development. But the little bit of development work that we personally have done so far, which is mostly, actually, there's a guy, David Travers, prof in the bio department, who's the one who pushed this, is using a zebrafish because it's easy to image into the zebrafish. Simpler, less vessel, you get a couple of ideas. But it's the cost is too high right now, at least for a little lab, right, to do this. That's why, you know, you need, I mean, if it's interesting, then all of a sudden somebody else will start competing with you and try to figure out a way to make it faster. And then the whole field improves, right? Okay, guys, I'm gonna go because I have a grant renewal that's due tomorrow. That's just the way it is, life in a big city. Okay, it's a lot of fun. And just, you know, you have any questions or anything, just email me or Zoom, as long as you show your face. I'm willing to talk to you.
Thank you, Professor Kleinfeld.
Yeah, okay.
Thank you, Professor Kleinfeld. I hope you come back. This was very informative.
Okay.
It was great to
Okay.
meet.
Yes, thank you very much.
Thank you.
All right, this was fun,
Thank
guys.
you.
Yeah.